Good afternoon, students. I'm very happy to invite for Zoom class. Today I'm going to introduce types of ovule and megasporogenesis, and third one, the development of female gametophyte. So first of all, let me take types of ovule. Previously, we discussed about the structure of ovule and the development of ovule. Right? Let us continue types of ovule. Before that, I'd like to once again review what is ovule. Ovule is the organ that is found in ovary. When you take a gynoecium, now let me draw the structure of gynoecium. This is the gynoecium. When you take a flower, at the center, you can find the female reproductive part of a flower that is called the gynoecium. This gynoecium consists of the upper side, you can see stigma, and the lower part is called ovary. And this ovary is a box-like organ that encloses ovule. Now here you can see the ovule is an organ that is found in ovary of a gynoecium in a flower. So here the structure of ovule is varied from one plant to another plant, right? Generally, ovules are classified into 6 types. The first type is generally ovules are divided into following types. The first type is the orthotropous ovule. The second type is anatropous ovule. First of all, let me give an example for orthotropous. I'm talking about the first type. This is a structure of ovule in which you can see, as I said, it is an orthotropous ovule. It consists of this orthotropous, ovule is a type of ovule in which it consists of the upper side is called microphyll region. So microphyll region is a place where the entry of a pollen tube which occurs, right? That is a microphyll region. It is the upper part of the ovule in case of orthotropous. The lower part of the ovule is called chalachal part. A chalachal region is nothing but it is the lower side of the ovule in which the central part is called embryo sac. So the embryo sac is placed at the center. This is called a embryo sac. Both this embryo sac and this chalachal region are protected by the protective structure that is called integuments. Integuments, right? So here in case of orthotropous, both microphyll region and the chalachal region are lying straight line. So both of them are located just opposite to each other, right? This type of ovule is known as the orthotropous ovule. So this orthotropous ovule is found in some plants. Let me give an example for orthotropous. That is Pipereaceae family. Pipereaceae family is nothing but it's a pepper family. It is also known as a pepper family. In the members of Pipereaceae family or pepper family, you can see the type of ovule is a orthotropous ovule. One more example I can say, there is a Polygoniaceae family. Polygoniaceae family. So there are two examples which are given for orthotropous. One is a Pipereaceae family, another one is a Polygoniaceae family. So what is an orthotropous ovule? Orthotropous ovule is a type of ovule in which both microphyll region and chalachal region which are lying just opposite each other, right? That is called the orthotropous. And next we pass on to second category, anatropous ovule. I am talking about second type, that is anatropous. Anatropous ovule is a type of ovule, but the shape of the ovule is inverted. Right? This is called it. I am talking about the second type of ovule, that is anatropous ovule. In which already, as I said, this is a microphyll region, right? Microphyll region, and just opposite you can see it's a chalachal end or a chalachal region. Both chalachal region and microphyll region which are inverted, right? Simply both of them are inverted opposite direction, right? But at the center you can say, as I said, embryo sac. And both of them are protected by, as I said, it is the integument region or integuments. So simply, in case of anatropous ovule, the microphyll region and the chalachal region are inverted, right, by an angle of 180 degree, right? So this type of ovule is found in some cases in angiospermic growth, that I can say. I'd like to give example for this anatropous ovule. In most of the dicotyledons and monocotyledons, see. Example for anatropous ovule. The first category is the dicot families. All the dicot members and monocot members. So both the dicot and the monocot. I mean, almost all the angiospermic group in which you can say anatropous ovule. Simply, I can say the ovule is inverted from the normal position, from the normal condition. Such a type is known as the anatropous ovule. Right? The next we pass to the third type. That is the hemianatropous. So here the hemianatropous is slightly varied from anatropous because the body of the ovule is simply it is formed, it is placed right angle to the funiculus. I'm talking about the third type, that is a hemianatropous ovule. This is a third one, third type, that is a hemianatropous. Hemianatropous means 50% from its normal position. I mean the microphylla region. This is a microphylla region. It's a chalachal end. Is the chalachal end and a microphylla region, or simply they are vertically? I mean horizontally. It is naturally this microphylla region and the chalachal region must be found in vertical direction. But in case of a hemianatropous, you can say the body of the ovule is placed just to the right angle of funiculus. Actually, funicle is nothing but it is the stalk of the ovule. That is the funicle. The funicle is nothing but a stalk of the ovule because it carries out the body of the ovule, which is very wider, not encloses microphylla region and its other region. Simply, I can say the hemianatropous ovule is nothing but the body of the ovule is simply placed right angle to the funicular structure, right? This is called the hemianatropous. The hemianatropous ovule is found in case of Primulaceae family. It is found in Primulaceae. Primulaceae family is nothing but it's a primrose family. Simply you can say primrose family. Primrose. It is a family, it is one of the member in which you can see hemianatropous, right? The next fourth category is campylotropous. Students, my dear students, we are discussing about various types of ovule. The fourth category is campylotropous ovule. It is very interesting one, campylotropous. Campylotropous, it's a type of ovule in which the microphylla region and the chalachal region are just opposite to the funicular region, right? But here in the structure, the body of the ovule, which becomes like a curvature. Body which becomes like a curving. Simply it becomes the means the shape of the embryo sac, which is slightly bends, and the one side of, I mean the egg part of the or synergist part of the embryo sac, which are connected to the microphylla region. Right? Simply I can say a campylotropous is a type of ovule in which the body of the ovule which becomes curving like structure. Now this type of ovule is found in Leguminaceae family, right? It is found in Legum family or Leguminosa family, or the bean members, pea plant, bean, right? In this category, you can see campylotropous, right? Or simply, you can say campylotropous is a type of ovule in which the body of the ovule, which becomes curving like structure. As a result, we can see the micronuclear region also bending down towards it is found, it is bending down just near the funicle. The funicular region or funicle, funicular is stalk of the ovule. Both is the funicular structure and the microphylum structure which are come close together, right? So this is called the campylotropous. It is found in Leguminaceae family or Leguminosa family. The next type is the fifth type. That is the Ambitropous ovule. We are discussing the types of ovule. Students, please remember. Right. This is a fifth ovule, that is Ambitropous. The Ambitropous ovule in which funicle is there. Funicle, as I said, it's a stalk of the ovule. And the micronuclear region, this micronuclear region and funicular, which are very close there, then that of campylotropous. As a result, we can see the body of the embryo sac, which becomes like a horseshoe shape, right? Horse shoe, a shoe of a horse, you can see, right? Because totally the shape of the embryo sac totally bending, right? As a result, which becomes like a horseshoe shape. So this type of ovule is found in only Alismidaceae family. Alismidaceae. Now, Alismidaceae is one of the members of Angiospermic group in which this Ambitropous ovule is found. What is Ambitropous ovule? Simply you can say the funicular region and the micronuclear region very closer to each other due to the development of a horseshoe shape of embryo sac. What this is called embryo sac, because embryo sac is found at the center, and by growing of this growing totally, it is growing in nature, and hence it is like it forms like a horseshoe shape, right? This kind of ovule is found in case of Alismidaceae family, right? The next last type is that is Succinotropous ovule. The last category, it is absolutely inverted more than once. That is called a Succinotropous ovule. We are dealing last category, that is Succinotropous. Succino, the term succino means coil-like structure. I repeat once again, the term succino means it becomes like a coil-like structure, because This is a funicular region. Funicle is a, and this microphylla region, the MC or MIC, microphylla region, both of them are lying the same direction as it is vertically. You can say the arrangement or the position of funicle and microphylla region, but it is a coiling, right? It is inverted. Absolutely, it is inverted, right? This is called the Sarcinotropous ovule. It is found in Cactaceae family. This type of ovule is found in Cactaceae. So the cactus is nothing like Casuarina or dry. The plants are grown in very dry region. It is a Cactaceae family in which you can see Sarcinotropous ovule. So far we have discussed 6 types of ovule, right? Orthotropous, Anatropous, Hemianatropous, Campylotropous, Amphitropous, and finally we discussed Sarcinotropous ovule. Right. Next we pass on to Megasporogenesis. Right. Next we pass on to a very interesting topic. So far I have taken only types of ovule, and it's time for discussion of Megasporogenesis. Megasporogenesis. Megasporogenesis is nothing but it is a process by which the orchisporial cell produces megaspore mother cell. Simply, it is a formation of megaspore mother cell. And again, this megaspore mother cell which undergoes meiosis cell division to form megaspore. Megaspore, it is the first cell of female gametophyte. This megaspore is gradually developed into female gametophyte. Or it is also known as embryo sac. Embryo sac. Embryo sac is also known as female gametophyte. This female gametophyte is derived from megaspore. The megaspore is produced by megaspore mother cell. This megaspore mother cell is produced from the orchisporial cell, right? This is a development of various stages of cells that is called megasporogenesis. I repeat once again. Megasporogenesis is nothing but it is a formation of megaspore from the megaspore mother cell. As a result, we can get female gametophyte, or it is also known as the embryo sac. Now it's time for a discussion of how this mechanism, how this process are taking place. There are several stages are there. The first stage is orchisporial mother cell. No, actually, this is an orchisporial mother cell. But orchisporium. Here, the orchisporium is found at the center of the cell. This is an orchisporium. Orchisporium is a group of specialized parenchymal tissue that is found at the center of ovary. You can see, right? This orchisporium gives rise to form 2 types of cells. The one is Primary parietal cell. The first category is primary parietal cell. And another one, sporogenous tissue. Or sporogenous cells. So there are two types of tissues are derived from the archisporium. This is the first development you can see, right? One is the primary parietal cell and another is a sporogenous cell. This primary parietal cell gives rise to outer covering. The outer wall of megasporogenesis is produced by primary parietal cell, whereas the sporogenous cells are responsible for the formation of embryo sac. I mean, the megaspore mother cell is derived from sporogenous cell. Now, let me give an example for this megasporogenesis, right? The second diagram. And third one, just I'd like to compare the diagram. As I said, this is a archisporial cell or AC that gives rise to from two types of cell. As I said, one is a primary parietal cell, PPC, you know very well, and second one is a SP, sporogenous cell. Yeah, this is a sporogenous cell which directly sometimes the last generation of a sporogenous cell may directly function as megaspore. Actually, this sporogenous tissue is derived from archisporial cell. These archisporial cells are originated from the nucellar region. The central part is called a nucellar region. Nucellar. What is nucellar region? It's a parenchymatous tissue. It is a platform for the development of archisporial cell. From the archisporial cell, primary parietal cells also originated and the sporogenous tissue is also originated. The last generation of sporogenous tissue may directly function as megaspore mother cell or directly sometimes it is converted into megaspore. Right? Megaspore mother cell. This megaspore mother cell which undergoes meiotic division. Megaspore mother cell. Meiotic division. That means during cell division. Meiotic is a kind of cell division that takes place in only reproductive organ of plant. Right? That is called a meiotic cell division. During meiotic cell division, or it is also known as a meiosis cell division, a single cell undergoes a division to form 4 cells, right? That is the nature of meiotic cell division. Among which there are 4 cells are produced from megaspore mother cell. Only one cell may function. Only one megaspore. It is possible to obtain. There are 4 megaspore from the megaspore mother cell. Only one cell function. Remaining 3 cells are going to be degenerated. simply the cells are not undeveloped, simply they are disappear after the development of only 1 megaspore. Particularly, I can say there are two regions already we have discussed the structure of ovule. The one is, as I said, it's a microphylar region in which megaspore is not functioned, it is not developed, right? Whereas in chalachal region, it is a region of chalachal. The chalachal region, only the megaspore is developed and differentiated into further division, right? The next stage, just I'd like to show you. After the development of single megaspore mother cell, it develops into another type of cell, right? The first division is meiotic division. I mean meiosis cell division. And the second division is the mitotic cell division. So please understand. The second division is mitotic division. Right, as I explained, among 4 megaspore in which the only 1 megaspore function and further differentiated into 2 types of cell. One cell is formed in microphylar region and another cell is developed in chalachal region. I repeat once again, the functional megaspore is differentiated into 2 nucleus. The one nucleus is pushed out towards upper polar region and the lower nucleus, another nucleus is pushed down towards lower polar region. I mean the chalachal region. How it is developed? By the appearance of simply a vacuole is suddenly originated here. Vacuoles. The sudden appearance of vacuole at the center in between microphylar region and the chalachal region at the center of the empty sac. You can see the development of microphylar cell and chalachal region cell. Again, this state undergoes mitotic cell division. Again, it undergoes mitosis cell division. Actually, the term mitosis is nothing but it's a kind of cell division in which a single cell which divides into 2 cells. When it undergoes cell division, it forms a single cell undergoes to form 2 daughter cells, right? This is called a mitosis cell division. And once again, mitosis is a cell division in which a single cell produces 2 daughter cells by the division, right, of karyokinesis and cytokinesis. That is called mitosis, right? Here, this is a 2 cell. Each nucleus of microphylar region and the chalachal region are further differentiated into or further divided into 2 nucleus in each pole, right? Both upper polar region and then lower polar, right? This is the upper side and then it's the lower side. So now you can see there are 4 nucleus, but the nature of cell is single cell. The nature of cell is a single, but the nucleus is only divided, not cytoplasm is not divided. Again, the development of vacuole, where at the center of this both upper and lower polar region of the embryo sac, again this vacuole is developed. Again, this 2 nucleus of polar region, upper side and the lower side are further differentiated into to form 4 nucleus. As a result, we can get 8 nuclei in a single cell. As a result, we can get 8 nucleus. This 8 nucleus are present in a single cell. That is called the embryo sac, you can see, right? So this is, as I said, very beginning, it's a microphylla region. And the lower part is a thalachelane. And at the center, you can see the central nucleus or secondary nucleus. Secondary nucleus, and this is a synergids. There are 2 nucleus which are occupied as synergids. There are 2 nucleus which are haploid in condition. In addition to that, at the center you find the egg cell, right? Egg is a female reproductive gamete, right? I repeat once again, egg is nothing but it's a uninucleated structure, but it is considered to be as a female reproductive gamete. It is going to fuse with the male gamete, right? Please remember. And the lower cell you can see polar cell or antipodal cell. So out of 8 nucleates, and each nucleate which are coming down and it is situated at the center, and one more gamete. So we have discussed in upper side there will be 4 nucleate, and the lower side that we have discussed in 4 nucleate. Among these 4 nucleate from upper and lower, you can see each nucleate from the each polar region. I mean, out of 4 nucleate, a single nucleate is coming and situated at the center, and another single nucleate from the antipodal region, right? The antipodal region are just fused to form as a secondary nucleus at the center. Now this is called a secondary nucleus. Now always a secondary nucleus from the structure we can find the secondary nucleus is always a diploid in condition. That is a 2-end state. Whereas the synergids are applied, and these antipodals are also applied, and the egg cell is also applied in condition, and synergy, etc. So out of 8 nucleated, you can see 3 nucleus are formed as a 3 separate cells, which are considered to be as a synergids along with the egg formation, which are found in the upper polar region, whereas in the lower polar region you can see 3 nucleated, 3 cell state, that is called the antipodal cell. Students, please remember, out of 8 nucleate, each nucleate from the upper side and the lower side, which are fused to form as a secondary nucleus. Now, this is the development of a female gametophyte. The next, I am going to show you the entire development of the embryo sac. The final touch, that is the final diagram. This is a female gametophyte. So far, we have discussed several stages. Right from the archisporial cell, we have discussed a number of cells, right? This is called a female gametophyte. This structure is known as the embryo sac. Just I'd like to explain very clearly, no problem, students. This is an embryo sac. Embryo sac. The embryo sac is nothing but it is a female gametophyte that consists of the upper side is called, when you take a cross section of the ovule or LS of the ovule, you can see the arrangement of embryo sac as it is. The central cell is called secondary nucleus. Right, and the upper side you can see synergids. There are two cells are associated to form as a synergids. In the synergids, you can find at the center, this is called a filiform apparatus. Filiform apparatus. A filiform apparatus is nothing but there is a finger-like projection which is found in synergid cell, which are essential for absorption of nourishment. I mean nutrition, water, mineral is absorbed by this filiform apparatus. Exactly, it is found in the synergids, at the center of synergids. You can see. What is that? Sometimes the question is going to be asked: which of the following absorbs nourishment in synergids? Right? That is a filiform apparatus. What are filiform apparatus? They are finger-like projection that is originated from the synergid cell. And just behind the synergid cell, you can see an egg cell. This portion is called as a egg cell. Now, egg is a male gamete. Egg is a single nucleated cell. It is considered to be as a female gamete, right? It is going to fuse with the sperm cell, right? And the lower cell, you can see antipodal cell. So antipodal cells are nothing but there are three nucleated and separated cells which are found in the lower side of the embryo sac. So simply, what is the nature of the embryo sac? The embryo sac consists of 8 nucleated but 7-celled stage. 8 nucleated and 7-celled stage. How it is possible? Because when we see, just when you look at the embryo sac, you can say there are 8 nucleus. 3 nucleus are found on upper side and 3 nucleus that are seen in lower side. At the center, you find 2 nucleus. So totally, on upper side, you can see 3 nucleus. At the center, you find 2 nucleus, and the lower side, you can see 3 nucleus. So totally, 3 plus 2 plus 3, almost 8 nucleated. But these 8 nucleated are enclosed by 7-celled state. This is very important area to be concentrated, right? So this is a way by which embryo sac or female gametophyte is developed, right? The next we pass on to simply how this embryo sac is developed. The development of embryo sac is various steps. Last minute, just let me take few minutes to discuss. So the development of embryo sac, it is possible to be developed in 3 ways. The embryo sac is derived by 3 methods. The first category is a monosporic condition, and the second category is a bisporic, and third one is a tetrasporic. The first type is a monosporic. The second type is bisporic condition, and third one tetrasporic. What do you mean by monosporic? Out of 4 megaspore, only 1 cell function. During meiosis cell division, it is possible to form 4 megaspore, only 1 megaspore function. That is called the monosporic. I mean the development of megaspore or the development of embryo sac from only 1 cell. That is called a monosporic. I can say some example for monosporic. There is a polygolum. The 2 megaspore cell, right? That is called a bisporic. A bisporic is observed in case of allium type. It is also known as the allium. A last category is a tetraspory. Tetraspory is nothing but the development of female gametophyte or embryo sac from 4 cells. During meiosis cell division, a megaspore mother cell produces 4 cells. Out of these 4 cells, which are involved in the development of embryo sac, such a condition is said to be a tetraspory. It is found in Peperomia family, right? Peperomia. So this is about simply types of ovule and megasporogenesis. So far we have discussed simple topic, right? My dear students, simply we have discussed it. This is a structure, right? Types of ovule. I repeat once again: orthotropous, anatropous, heminotropous, campylotropous, ambitropous, and succinotropous. For each category that I have given number of example. When you take any ovule, you can see microphyll is upper polar region, and the zalagel is the lower polar region, and the center you can see embryo sac. It is protected by the protective layer is called as an integument, right? This is the integument region that covers this embryo sac. And we have discussed anatropous. It is found in Malay, dicot, and monoconidae. Then heminotropous. It is found simply in primulus or primrose. Then campylotropous that we have discussed. Then finally we discuss the succinotropous. Then megasporogenesis that we have discussed. What is megasporogenesis? It is a development of megaspore from the megaspore mother cell by several cell division, right? Because it is initiated from the organsporial cell, in which you can say sporogenous cell, and one cell on the other side primary parental cell. These sporogenous cells are responsible for the formation of simply megaspore mother cell, right? These megaspore mother cells are developed by several cell division, meiotically and mitotically, right? And next, the vacuole is also originated, and due to the development of vacuole, both the upper side and the lower side of the embryo sacs are pushed out, and finally it gets eight nucleated stage, right? Among these 8 nucleated stage, you can say the synergids along with an embryo, you can see 3 nucleated. And at the lower side, you can see antipodal region that consists of 3 cells. At the center, you find secondary nucleus, there are 2 nucleated. So totally, the embryo sac is contained by 8 nucleated 7 celled stage. That is simply it is known as a female gametophyte. And the development of this female gametophyte by 3 ways. One is monosporic, another is bisporic, third one is tetrasporic. Monosporic means the development of embryo sac from this single megaspore mother cell. Whereas in bisporic condition, the development of embryo sac or female gametophyte from 2 megaspore mother cell out of 4 cells. Generally 4 cells are produced, right? Now talking about a tetrasporic mother cell, out of 4, these 4 megaspore mother cell are also involved for the development of embryo sac. Is found in Peperomia, right? It's time for interaction, students. Anything else? I'd like to interact with you. How did you understand, students? The last topic that we have discussed. Students, anybody can answer? My dear students, could you tell us in few words what is monosporic? Students? What now? Will you give an example for monosporic?
Only one megaspore function.
Very good. Only one megaspore is involved. Very good. Keep it off. And what are the parts are seen in ovule? We have discussed a number of ovule. This is a structure of ovule. Which one of the parts are seen there?
Yes, Swati, answer us.
What is the first type of ovule?
Orthotropous ovule.
Very good. Orthotropous ovule. Okay. What is the second? Will you give an example for anatropous ovule? But did you understand what I taught you today?
Dicot and the monocot.
Very good. Very good. Dicot and monocots. And the next, what do you differentiate a hemianatropous from anatropous? I have drawn two structure. One is the anatropous and maybe anatropous. How do you differentiate, ma? Could you tell us in few words? Diagrammatically, you can make a difference.
Anyone?
Anyone? Right. Can you give an example for such an anatropous ovule?
Cactaceae.
Cactaceae family. Very good. Very good. Keep it up. Okay, what are the parts are seen in embryo sac? Because this is a finally we discussed it. It's a female gametophyte. In which one of the parts are seen there? Students are able to hear? The beautiful structure is seen on the screen. I have drawn and labeled some parts. What are the parts are seen there? An upper side. Okay, how many nucleated in condition? How many nucleus are there?
8 nucleated.
8 nucleated and 7.
7 cells.
Thank you for joining, and it's time for giving assignment.
8.
Right. Draw the structure of embryo sac and label the parts. Right, please take down some assignment. I'm going to give you. Please take. Some assignment I'd like to give you. The first question. Draw the structure of orthotropous ovule. And label the parts and classify or mention the types of ovule in plants. And third one, explain the development of embryo sac. Explain the development embryo sac. Right, this is the assignment part. Please take down. Right, thank you for joining, students. If you have any clarification or doubt, please send it as a question to your respective subject teacher. Right, thank you for joining.